Hi, my name is Shreyas Hirway, and my project is titled A Novel Computational Machine Learning Pipeline to Quantify Similarities in 3D Protein Structures. To provide some background on this project, animal models have been widely used in drug development to understand target biology and assess the pharmacology and safety in studies before human clinical trials can be conducted. Therefore, choosing the correct animal species for analysis is a crucial part of the process. Commonly used practices for species selection involve using legacy species based on years of previous usage, target biology, species availability, or other practical considerations. However, in the absence of a relevant species, comparing the target sequence and structure homology between the human and other animal species is a common first step in the process, as the lower percent homology can eliminate potential species that would have a lower pharmacology activity. Therein lies the challenge with this approach. The 2D comparison solely focuses on the pairwise sequence or structure rather than looking at the more biologically relevant information available between the proteins. Therefore, to bridge the gap in the field, we propose an end-to-end computational pipeline to quantify similarities between proteins and use that data to identify the animal protein that is the most similar to its human homologue. In this study, we gathered human and animal protein targets from structural databases such as the ISMB protein databank and the Campbell bioactivity dataset. Using the protein databank data, we generate attributes based on the structural comparison as well as the nominal annotations for the human and target entries. The bioactivity data, specifically the IC50 values of the targets, are used to generate similarity scores that will then benchmark the human to animal comparison. Finally, all of the generated data and scores will be utilized in a machine learning model, which will determine the optimal animal homologue for a human protein. The flowchart in the middle represents the entire computational pipeline. It can be separated into three different modules. First is dataset generation, followed by machine learning, and ending at model assessment. There are a lot of components in this flowchart, so let's break it down piece by piece. Firstly, we have the dataset generation module. We begin with the protein data processing step, which involves parsing through the Campbell data, which is a dataset of protein entries that contains bioactivity data, such as species, assay, ligand, and other information for each of the entries. So we start with a total of 6 million entries of data for human and animal target proteins, and begin filtering down based on bioactivity data, which in our case is the IC50 values. Next filtering step is the assay description, followed by ensuring that the same ligand is used for the human and the non-human entries, leaving us with a final set of 3,700 total entries. These entries are then used to generate a protein data bank, PDB-based dataset, which will directly list a human target entry next to its non-human homologue. From the Campbell database, we retrieve the UniPro database ID and convert it into a PDB ID, which will then be important later. Now, the IC50 values mentioned earlier will be used to generate similarity scores for each human to non-human entry comparison. Next, in the machine learning module, we will focus on feature generation, which consists of two different parts. First is the structural alignment metrics, where we use a structural comparison package to generate metrics focused on comparing the human protein to the animal protein. Next, we perform natural language processing on annotation-based data, such as the protein family, cellular process, molecular function, etc., for each of the protein entries. Based on these metrics, we generate two different datasets. First is the RCSB dataset, which consists of a giant database of 3D protein structures. And the second one is the AlphaFold 2 database, which consists protein structures predicted by Google's DeepMind AI program. Now, for the machine learning process, we perform regression analysis using the XGBoost model, where we first identify the most important features in the model and then conduct a parameter study to fine-tune the model parameters. The final output of this regression model will be predicted similarity scores for each human to animal structure entry. Now, in order to assess the model, we first take the PDB IDs and convert back to their original target name. Then we perform majority ranking, where for each human target we compare the similarity scores between all of the animal entries for the target and select the animal species that is the closest to the human score. Based on that, we select the optimal species for each human target. Now that we've predicted an animal species for all of the targets, We compare these to a real-world case study of experiments where the targets, phenotypes, and animal species used were documented. So we find common targets between our predictions and this case study set and check if our predicted species is the same as the documented ones. And ultimately, that determines the accuracy of the model. Now, for the results that we show here, we went one step further and looked at the phenotype that's associated with each target. And if you group these targets together of the same phenotype, using majority ranking, we can ascertain the optimal animal species that is used for studying a certain phenotype. For the three phenotypes shown, dry eye, hypotension, and anemia, mouse models are prevalently predicted and are in agreement with the real-world dataset, thus demonstrating our model's ability to determine the optimal species to study a certain phenotype or disease as a whole, based on the individual targets relevant to the condition. So to provide a quick summary, in this study, we built a computational pipeline that uses structural and nominal data to quantify tertiary protein structure comparisons. We were able to use the IC50 bioactivity data as a means to benchmark the protein similarity between the human and the animal targets. And we matched our best model's predictions to a real-world data set with a high level of accuracy. This pipeline will be able to automate the animal species selection by selecting the species whose ortholog protein is the most identical to humans. Now, there were some limitations that popped up during the study. For instance, we had a small set of comparisons for each target, but this can be remedied by increasing the number of animal species that are available for each human target. In the future, other applications of this pipeline can include off-target safety assessment. Thank you for your time, and please reach out if you have any questions.